All right, so today I'm going to talk about, actually just quickly review motor proteins, topic 3 and topic 4 on fluorescence. And then the main new topic will be topic 8 on DNA structure and two-state systems using DNA as a model system. I'm skipping topic 5, 6, and 7 from my lecture notes. You can actually look at my lecture notes to read, more details, because there'll be guest lectures, not next week, but 2 weeks from now. 2 lectures I'll be out of town, and so my postdoctoral fellow Jingyi Fei will give 2 lectures. The lectures will be shorter than usual, and on 2 mainstream fluorescence techniques that biophysicists use. One is called FRED, the other is called super resolution imaging. And then you'll be also visiting my labs to attend lab demos. So the class will be like half here and half in the lab twice. Sounds interesting? Okay, don't break anything. I'm going to charge whatever the damage to your advisor's account. Okay. So I want to discuss 3 different things about the motor proteins. What's this? Size. Well, the motor proteins move on biomolecular tracks made of polymers, and they because they are the tracks are made of single monomers. There is actually a smallest unit you can imagine, so it's not continuous. The track is quantized. So in a sense that the step size of a motor is not indefinitely small. There is a finite step size the motors can take. And speed of the motor on the track is how quickly it moves as a function of time. So it'll be defined as micrometers per second type of unit. And processivity is how far the motor can go before falling off. So step size can be measured if you're able to determine position x of a motor protein as a function of time, and you measure the position as a molecule is moving, let's say, in the plus x direction, and then you follow the position as a function of time, you will see some trajectories like that. And of course, this is highly idealized. Okay, in reality, there's a lot of noise. And if the step size is big, then the noise can be negligible. And then you ask a random stranger on the street, the person can tell you, okay, there is a step, and so on, right? Everyone will agree. But if the step size is not very big and noise is large compared to the step size, then maybe you will see that there is a step, but maybe the only other person in the world who will agree with you will be your mother, right? So in some cases, you have to rely on the computational tools to really show that there are really discrete steps. Because if you rely on your visual inspection, sometimes it can be misled. But anyway, if you assume that you can determine each step based on the data, then you can measure each step, delta x, and you can measure it for all of them. And then plot a histogram. So this is delta x value, and then number of event. And then, so if you're lucky, I mean, you'll get a delta function, but usually that's not the case. You'll get a histogram that, follows some distribution. And from this, the mean value will be the step size. Okay? So this can be eight nanometer for kinesin, and 37 nanometer for IC5 and so on. You can also measure the dwell time of each step. Okay. And ideally, what you get here is, if you plot the histogram again, number of events. It's going to show an exponential decay. Following this rate, and here the k will be the stepping rate. Okay, given in the unit of second minus 1. So if you want to think of it this way, let's say this is the x direction, and you have, lattice location n-1 position, nth position, n+1, n+2, and so on. So you can take the step transition here. That step occurs with the rate of k. This one takes, and so on. Okay, that's what I mean. And each step is stochastic. It follows the exponent. So this is called the Markovian process, because as long as you are here, it doesn't matter how long you've been staying in that state. In terms of determining whether you will take this next step in the next time unit or not. So history independent process is the Markovian process, and in this case, the histogram of delta t should be a single exponential. Questions? But let's imagine a kinesin molecule or myosin 5 molecule walking like this, and then imagine that I have a light bulb to my shoe, like on the front, and you're looking at me from, ahead of me. So here you cannot see my light bulb because it's hidden by my left leg. And then when I step forward, you'll see my light bulb, right? So you can measure when that step is taken, okay? So that if you actually determine that moment of motion just based on when the right foot moves forward, then that doesn't happen every time. It actually happens every other time, because when my left foot moves forwards, it does not actually give you the light actually suddenly appearing, right? So if you then in that case, you'll be actually measuring time it takes to go from n-1 to n+2, and then also to n+4 and so on. So if you measure the time every time my right foot moves forward, I don't measure it, and I measure it again when my right foot moves forward. Then and they plot the histogram of that dwell time. So in that case, your delta t histogram actually looks kind of like that. It's not exponential. It has a lag phase and a peak and then decay, and it actually follows a function that looks like this. That's because you cannot take two hidden steps simultaneously, right? Because they have to occur in succession. So the chance that they will happen simultaneously is very small, is 0. And that's why you get this function. This is generally called a gamma function, gamma distribution. In general, if you imagine that instead of having 2 legs, imagine that I have 3 legs, okay? And then this is the one that has the light bulb. And then the next step, this guy detaches from the rear and then moves forward to go here. And then the next step, and that one detaches and then moves to the front. Then this guy will go to the middle, and then the next one it'll go to the back, and then and this can continue. Do you know what I'm talking about? So what I would call this three-legged kinesin. So imagine there's no such thing, but let's imagine you have something like this. Then you measure actually time between this step and then this step, right? If you want to measure this dwell time, and because you get signal only when that happens, let's say, then this should actually involve 3 hidden steps of equal stepping rates. Then the distribution will look like that. In general, if you have 4 n hidden steps, so this is the case of n equals 3, and this is the case of n equal 2. Then when you have n hidden steps, that distribution will look and you can show this by doing a simple calculation, convolution, doing it multiple times. So for a large n, that histogram can appear to be really sharp. Okay? And in fact, by looking at how sharp that is, you can deduce how many hidden steps there must be. Okay? Now, the speed. Any question about this? Is step size times the rate. That's the speed. And typically for kinesin, about 100 steps per second. So that is the rate. Per second. So the speed of kinesin would be 8 nanometer times hundreds per second. So the rule of thumb is that typically about micrometers per second for many of the monoproteins moving on microtubules or actin filaments. An important number is this one. So there is a reaction, the ATP hydrolysis reaction that is happening 100 times per second. So that means each reaction takes about 10 milliseconds, right? And this is a very typical number for many of the reactions in biology. Some chemical reactions important for biology happen, every few milliseconds. Okay. So it's hard to find anything that is much faster than a millisecond, and so this is quite a typical number. Okay. So you want to get a feel for that. For example, it's hard to simulate a protein dynamics for as long as a millisecond, right? So what that means is that if you want to use a brute force computer simulations or Latin simulations to understand how these motors move, then you still have to wait for probably another few years before you can actually do it, because these reactions are relatively slow, takes milliseconds and so on. But obviously, that is fast enough for us to survive, right? Processivity. So in terms of this terminology, there is no consensus definition. I mean, everyone knows what it means. That it's basically how far it goes before falling off. So some people use this term to denote one definition. Definition number one would be, okay, the average run length. So when kinesin binds to the track, and how far does it go before falling off? Okay, and it turns out that a kinesin takes about about 100 steps before falling off. So then the average run length will be 8 times 100 nanometers, 800 nanometers. So about a micron. Just to give you a sense, basically, it moves about a micron and then fall off. So that's actually not so good if you think about it. If you want to send an object from somewhere here to a motor neuron down here that contacts the muscle, then that's actually much longer than a micron. So clearly it does not bind and then keep walking without falling off until the end. It falls off at some point. And the question is that why? Why does it have a limited processivity? There can be many reasons. It's possible that usually the track is bound by many different proteins, so it's congested. So if you insist that you must go on this path, right, then you may be stuck permanently. But if you become a bit more flexible and then, okay, let me just come off a little bit and then move around and then rebind somewhere else, then we have a chance to bypass that obstacle and then make its way. And that's actually how to become a successful grad student in PhD research, right? You don't want to have a narrow view, okay, this is the goal, and then I shouldn't do anything else. When things don't work, take a break, and then something else will work, and then you can get a PhD. So it's good to have a limited processivity when you do your research. All right, so that's one definition of processivity. So people sometimes call processivity of kinesin is in 800 nanometers. But I would call that a pretty loose definition. And another more rigorous definition is that probability that it instead of falling off. So So according to this definition, then the processivity P will be 0.99 for kinesin, because it takes about 100 steps before falling off. Some people use a term highly processive, when this number is high. But even if it takes only 2 steps before falling off on average, it is still processive, right? Questions?
So, how does it fall off? Is there any mechanism in the bonding with the specific root?
Well, this is a very, very good point. So, we have 2 molecules that can bind each other, but they can also come apart, so they can dissociate. It's driven by thermal fluctuations, and in a sense, no interaction is permanent. It depends on temperature. It depends on the interaction strengths. It depends how good a match there is, and when you try to find a perfect match, girlfriend or boyfriend, you have all these transient interactions, and but everything.
It's thermal fluctuation.
Yes, yeah. So if you form a complex, and then if you then freeze that into, if you drop the sample to liquid nitrogen, they're not going to fall apart, right? Any other question? Okay, so let me move on to the fluorescence. I showed this to you on Tuesday. This is called the Jablonski diagram, and is the electronic level structure of fluorescent dye molecule. In fact, whatever coloring reagent that people use is also just like that. Okay, they take up light, and take up light of certain color. That's why your clothes has certain color. So you start from the bottom of the ground state, and then you take up a light, a photon of energy h times nu1, Planck constant times the frequency of the light, and it goes to somewhere on the excited state manifolds, because in addition to the excited electronic state, you have additional vibration and rotational states, forming essentially continuum. And then once you go there, then it can come down here within 1 picosecond. It relaxes down to the bottom of the excited very quickly. Takes only about a picosecond, 10 to the minus 12 second. And then it sits here for a long time. And then eventually it comes down to the ground state after about 1 nanosecond. When I say a nanosecond, I'm talking about the order of magnitude. It can be 0.2 nanosecond can be 5 nanoseconds and so on. Okay, depending on what you have. And then this will come down to the ground state within, again, 1 picosecond. So most of the time is spent in the excited state. Okay? Now, because of the energy lost here and here, okay. So if you want lambda 1, which is so energy is different between the excitation emission photons. So this is your laser light energy. This is your fluorescence energy. Energy is larger for your excitation than the fluorescence emission. Therefore, the wavelength of the light is shorter for the excitation light compared to the emission light. Okay, and this effect is called Stokes shift. Okay, so energy difference. So if I had to plot the spectrum of absorption and emission of a fluorescent dye molecule, then it'll look like that. Wavelengths. That's for the absorption spectrum. And it's broad, typically tens of nanometers across in the visible light because, again, of the vibration and rotational states. And then emission spectrum looks more like a mirror image. Okay. And this is the emission spectrum. And the peak wavelength is shifted, and that's called, right? That's a Stokes shift. And this can be between 20 nanometers and 80 nanometers, depending on the molecule that you're looking at. Okay. So what's nice about the fluorescence is that if you say use a laser, this is a laser light of this wavelengths, then you can excite the molecule pretty well. Okay. But then your emission is actually much more red in terms of color. Okay. So if you have a good filter to reject a certain wavelengths, then you can collect only the fluorescence, without being bothered by the laser scatter light. And that's very important because otherwise this can be 1,000,000 times brighter than your signal if you're looking at single molecules. Now you can buy these filters for about $100. So you can just go to chroma.com, omega.com, and then use a credit card to buy. So it's expensive for students, but for research, it's okay. So you can easily just reject the laser light and collect only the fluorescent signal. That's the really beauty of fluorescence. So essentially background-free measurements. Questions? Yes.
Are the line and the dotted line symmetrical on purpose, or is that just on accident?
They are more or less symmetrical, and that's because when you go up, you can go. This is the main thing, right? But you can also. This is the main band. When you go from here to here, that is the main band. We can also go to even higher energies, right? Because of additional energy level. So that's why you get this additional shoulder here, right? But when you come down from here to here. That's the main transition, right here, right? But then you have additional transitions that are allowed that have lower energy, or longer wavelengths on the shoulder.
Oh,
Okay.
now I see. Okay, thank you.
But that doesn't explain the probability. Why is it symmetrical?
Oh, in a sense, that I think explains this for the diagrams I draw, right? So basically, it makes sense as if you accept the fact that they all start from the bottom of each electronic state. Then when you go up here, because additional states are always higher in energy. So compared to the main transition, everything is higher in energy. But when you come down, compared to the main transition, everything else is lower in energy. So you have a main transition here, and then lower in energy, or longer wavelengths.
And also, did you draw the shape intentionally, or just this shape?
Oh, this is a real shape. So this is actually main electronic band, and this is actually additional main vibration state. Okay. So if you don't have any rotational state, then you see, two peaks. In fact, if you cool it down, if you measure this in a crash temperature, then you'll see two main peaks. Questions? So you may ask me, why am I using a laser light at these wavelengths instead of exciting at the peak wavelengths of the absorption? The reason is that because fluorescent photons are precious, you don't want to waste them. Okay. Now imagine that you're trying to excite it right here. Okay. Then you need to use some kind of filter to reject this laser light. But there's no perfect filter that blocks only that wavelength. So you will actually lose some of this signal, because if you're trying to block here, then you lose some of this signal here. Okay, so you will actually sacrifice much of the fluorescent signal coming out, right? But if you excite it here, then although you don't excite it as much, but you can compensate it by actually just cranking up the laser light. But then you don't actually have to worry about losing fluorescent photon. You can collect almost all of it. Okay. So if you do the cost-benefit analysis, you can do actually much better by exciting a little bit to the left, and then collecting more of what's coming out. And this actually is relevant to photobleaching. So if you leave your shirt out in the sun, I guess sometime later, then the color fades. That's called photobleaching. And in fact, fluorescent molecules undergo the same thing. So photobleaching is, it's an oxidation reaction. So if you shine, the molecule goes to the excited state, then somehow it reacts with oxygen molecules in solution to become a different molecule, right? And then that is no longer colorful. That's photobleaching. So in a sense, the reason why it's so precious is because you have a limited number of photons you can get from your fluorescent dye before it photobleaches. So you want to basically maximize the collection efficiency. Photobleaching has one actually nice feature, for a scientist. So let's say This is your fluorescence intensity you're measuring from a single fluorescent molecule as a function of time. Then you see some signal coming out of it, with some noise. And then suddenly it drops to the background level, because at this very moment, the molecule actually bleached in a single step. So if you're really looking at a single molecule, then photobleaching occurs in a single digital step. Okay? Whereas if you have many molecules, then the signal will decay gradually, exponentially. Okay? So that fact can be used to show that yes, indeed, I'm measuring a single molecule instead of ensemble of molecules. Second reason why this can be useful is for stoichiometry determination. So do you know what that is, stoichiometry? What?
The ratio of the different constituents?
Yes. So, some proteins actually do not function as a single protein. They have to assemble into what's called oligomers. Okay, dimer, trimer, hexamer, pentamer, and so on. So, in fact, actually, a majority of the proteins found in nature are actually assembled into oligomers. And dimer, primer, tetramer, so two proteins can form a complex, a dimer, and they can form a trimer. They can form a tetramer. What's next? Pentamer. Hexamer. And what's next? Heptamer. Great. Next?
Octamer.
Octamer. And nonamer. Decamer. Okay. And? Undecamer. Okay.
Dodecamer.
Dodecamer. Okay. And? Okay, that's the limit of my knowledge too. But I think actually all of them actually exist in nature. Okay. Actually, the only thing I'm not so sure about is the undecamer, but everything actually exists in nature, and it's very controversial. Okay, some people actually fight. People argue and they yell at people in the conferences. You're wrong. It's a dimer. No, I'm right. It's a tetramer. Whatever. Right? So, and in fact, in biology, controversy is much more common than in physics because I think two reasons. One is that usually measurements are not as precise, so open to more interpretation. Second, people study many different systems, right? Some people study proteins from E. coli. Some people study proteins from human, and even for the same system, some people do measurements in vitro. You know what in vitro means? In vitro means in glass. So basically, you're measuring things, outside the cell. Some people measure things in vivo inside the cell, and so the conditions can be very different. So, but I measure something from like frog protein in my test tube, and then I say it's a trimer. But trying to make my study more important, right? I make a claim that this must be true for every organism, right? And then someone else claims that I measure this from fruit flies, and then it's different, and so that's another reason why they can be controversial. But this stoichiometry is actually really quite controversial because first, it's not so easy to say where it forms a tetramer or pentamer. More importantly, it's not so easy to say which one is the more actually functionally relevant one, right? And so that's another controversy there. And the measurement itself can also perturb the system. By attaching some fluorescent probes to these molecules, you may actually change their properties. So it's really hard to get a really conclusive answer. Now, the reason why I'm talking about this is the following reason. So if you have a fluorescent molecule attached to each protein, like fluorescence here, and same here, and so on. And then you isolate a single protein complex, and you measure fluorescence signal as a function of time. And this will show two-step photobleaching, right? So you just count the number of steps. Okay, I see 2 steps. It must be a dimer. And here I see 3 steps. It must be a trimer. And here I see 12 steps. It must be so that is something you can actually do. But in reality, it's not that simple because the labeling efficiency cannot be 100% all the time. So if you only label 90% of the molecules, then some of them are dark, and you have a distribution instead of having all of them showing 2 steps, for example. So you have to actually do some statistical analysis to put a bound on what you can claim. Questions?
Can you please repeat the last stepwise what you said? So if we are measuring the fluorescence in a dimer, so you're saying that we will be seeing it in the form of 2 steps?
Yes. Photobleaching, yeah.
Photobleaching, because there are 2 molecules.
Yeah. But if the labeling efficiency attaching a fluorescent molecule to a protein is not 100%, right? Let's say 80%, then some of the dimers will have only 1 fluorescent molecule. Yeah.
Okay.
Some of them will have actually even 0, right?
Okay.
So some of the molecules will show just 1-step photobleaching. Okay. Then you have to then use binomial distribution and so on to pull out the information. Yes.
Yeah, I get it. So how do we realize which is the first step and which is the second step? I mean.
Oh, in that case, you see that as a function of time. So when you see 2-step photobleaching, I don't know which one is bleaching first.
Yes.
I don't know that, right?
Okay.
Yeah.
So they don't. Okay. So they obviously don't bleach simultaneously. So we just see.
Okay. Sometimes they can because of course you can. This can happen, right? So that so you have a small fraction of events that occur with single step. Then, but in that case, intensity that you measure will be twice as big, right? So that's another thing that you have at your disposal. So there are actually many new things you can do if you go down to the single molecule level. In a sense, it's like quantum physics. You have the smallest unit, and then you can do things that you'll not be able to do. Okay, so then I want to talk about polarization effects. All right, so that's a fluorescent molecule. Actually, I don't know what it is. I just made it up. But here you can actually define a dipole. For absorption of the laser light. So when you and what it means is that relative to the molecule itself, there's a defined direction. And if you come with a laser light polarized, if the laser polarization is along this direction, you can excite this maximally. But if you come with a laser light that is polarized in this direction, you have a minimal zero excitation. So if the direction of the laser, I don't know which symbol to use. It's just N. That's the polarization direction of the laser light. Then your intensity that you measure will be proportional to the square of the inner product. Or if you want, if this is that laser light polarization direction and this is the angle theta, then this will follow cosine of that angle square. So if you rotate the laser polarization as a function of time and measure a single molecule, then you'll see a signal that is going up and down in this manner. So let me show you one slide. Oh, okay, so it's not automatic, okay. So when I was a student, I had three opportunities to use writing system called LaTeX. Do you know what that is? How many of you have used it before? Okay. So I use it precisely 3 times because to submit my papers to physical review letters, I had to use LaTeX. And each time I learned it and I forgot it and I relearned and forgot and so on. I succeeded twice and then failed once, so that paper was published elsewhere. Let me show you one figure from my first PRL. Okay. What's going on? Yeah, let me just start it first. Yeah, I don't want to show you something unseeming or unsightly to you guys. Yeah, so. So that's actually what I'm talking about right here. So what I did was I placed a single dye molecule on a glass surface, and then I came with laser light that is rotating in polarization. So I had an electro-optic modulator that I controlled using my program. So you can actually change the laser polarization like this as a function of time repeatedly. And then I measured the intensity of fluorescence from a single dye molecule. You see nice oscillation. Actually, really comes down to essentially 0. Okay? And you can actually fit the data with the cosine square function very, very nicely. Once you have average, and you can determine this angle very precisely and so on. So I was very, very impressed with my ability back then. Yeah. again, you would not be able to see this if you have a randomly distributed molecules in ensemble, because then they all point in random directions. Then you will just see a flash signal, right? Now, this can be used for real biological applications. And let me give you an example here. So this is the actin filament. Myosin molecules move on the actin filament, but you know, you can imagine the myosin molecule standing up and simply walking straight up, but filament itself has a helicity. So another possibility is that as it moves, it follows the helical path to rotate around the filament. And it wasn't clear how that works. So what these people did in Japan, I think this is another thing that you will, no, you're not going to look at that anyway. What they did was they, instead of putting the actin filament on the surface and letting the motors move on the actin filament, they put the motor proteins on the surface, upside down, and then they can actually push the filament in one direction. And so in Korea, actually, maybe 30 years ago, there was a period when the dictator rounded up all the so-called gangsters, you know, young gangsters on the street. They put them into military camp for 6 months or whatever. And then what they did was every day they would carry a big log. You know, several of them would, you know, push the logs around just to, you know, repent and then whatever. So just like that, you know, these motors can actually carry, you know, push the filament in a directional manner. So what they did was they attached fluorescent dye molecule to the filament with a fixed orientation. And then initially, let's say it's pointing in this direction. And then if you measure the fluorescence signal in two differently polarized channels, vertical and horizontal, and when you have two dye molecules, in the vertical channel, this will be bright, but in the horizontal channel, this will be dark. I mean, this will be dark in the vertical channel and vice versa. So as it rotates, and then this guy, if the filament rotates, then this angle will change. And then that means that as a function of time, you will see the intensity actually going up and down. So can you turn off the light? Maybe I can show this to you better. So let me just show you one example. So this filament is moving. There's a single dye, and that appeared mostly in the horizontal channel. And then later on, now it tries to shift to the vertical channel, and then comes down to the horizontal channel. Okay? This is because as the filament moves, it is rotating to rotate the dye molecule's orientation. So from this measurement, they could basically measure how quickly it rotates and show that actually rotation is actually much slower than the pitch of the filament itself. Can you have the light on? So fluorescent polarization can give you actually a lot of information about, you know, what's going on, and so it can be a very powerful tool. All right, the last point I want to mention before we move on is that, you know, so far I have told you about organic dyes. And you know, Jablonski diagrams really apply to these dyes. Okay, but there are two other main actually fluorescent object that people use, and one is called GFP. And the other is called quantum dot. And GFP is a protein discovered in some species of jellyfish. And what's nice about this is that because it's a protein, you can actually encode that information into DNA. And if you then put that piece of DNA, you know, in front of a gene of interest, then in your chromosome, then you can see the fluorescence whenever that protein is made. And you can also see where that protein is inside the cell. And so you can do a lot of cool experiments. So this really revolutionized biological studies. And as a result, 3 scientists won the Nobel Prize 4 years ago. Okay, and what? Can you guess which prize?
Chemistry.
Chemistry, right? Because chemists are not that smart, right? They start giving out the prizes to biological discoveries. So whenever there's a big prize-worthy discovery that does not quite fit the physiology prize, then they use the chemistry prize. My prediction is that there will not be a physics Nobel Prize in bio topic for another 100 years. Yeah. But you can get the chemistry prize. So quantum dot is what used to be called nanocrystals. These are quantum confined structures in all 3 dimensions. So they are like dots, typically just a few nanometers in size. And you can even buy them commercially in a manner that can be attached to biomolecules. And what's nice about them is that they are much brighter than organic dyes. And they are also much more photostable. They don't bleach very quickly. And but there are 2 downsides. One is that they blink. So instead of giving you a steady fluorescence, they turn on and off randomly. And in fact, the statistics follow the power statistics. So it's really quite lousy if you want to use it as a light source. Second, they are quite large compared to fluorescent molecules. So they have a much bigger chance of perturbing the system. And they limit, and it's hard, and as a result, because of the size, it's harder to get them into the cell and so on. In fact, when I was finishing my PhD, some people asked me if I wanted to join a company to commercialize the quantum dot technology. And I almost did, but then they decided I'm not good enough for them. So I didn't. And the company actually started, and they raised millions of dollars. But then they had to sell it for pennies because there was not a big enough market. But that product is still being offered by a major bio-reagent company. All right, let's take a break for 5 minutes, and then we'll then switch over to the next topic of DNA and DNA, 2-state systems. Okay, let me start. So I want to talk about DNA. DNA is a famous iconic molecule. This is a place structure in a place called Lawrence Hall of Science, right up the hill of UC Berkeley campus. And some people like DNA enough to have a tattoo. And you can climb up this strand of DNA to see where life takes you. You know, popular cartoons. And this is a really famous picture of Watson and Crick. Do you know who is Watson? And who is Crick? The younger guy is Watson. He was 10 years younger. Okay, who is Watson? He has less hair, right? So that's Crick, Watson. And that is a structure they built to model the DNA. So nowadays, you know, when people solve the structure of a biomolecule based on X-ray diffraction, they start usually de novo. You know, you basically look at all kinds of Fourier transform and then determine the phase and so on to solve the structure. But what they did was different. They tried to think about possible ways of arranging the DNA so that you know, in the Fourier space you will see the diffraction pattern, right? So that was kind of, you know, okay, let's see if that works. And so make something and then does that actually give rise to whatever this diffraction pattern? So that was a very old approach, and they, you know, succeeded in a relatively short amount of time. So that is 1953 paper from Watson and Crick. And so in this paper, Francis Crick's wife, she drew the figure on the kitchen table. So back then they didn't have Adobe Illustrator or things like that. So it was, she just drew it, yeah, right? And it actually is amazingly accurate. Right? And you have two strands forming a right-handed helix. In fact, there's a website, I forgot the name, but it's a collection of images that people produced of left-handed DNA. So, you know, many people make left-handed DNAs by mistake. You know, basically when you try to draw DNA for publication or animation, whatever, you can make a mistake. In fact, the Nature special issue on DNA structure, DNA sequencing, human DNA, genome sequencing in 2003 or 2002, that issue also had a left-handed DNA. So there's a website collecting all of the mistakes. Anyway, so but she didn't make a mistake, and you have these base pairs, and each base pair is separated from the next base pair by one base pair. Well, okay, 3.4 angstroms. And this paper ended with this sentence that it has not escaped notice that the specific pairing suggests a possible copying mechanism for the genetic material. Okay. So this is how you can copy the genetic material. And many people believe that this is the understatement of the century because it's such a modest statement, right? So in fact, I read a book by Watson. I think I mentioned this, you know, the Gamma of Girls and Genes, whatever. So he says that after they published this paper in April that year, they got worried that someone else may actually propose this in a more explicit manner and take the credit for that discovery. Right? So they wrote another paper and sent it to Nature, published the next month. It's actually quite interesting that back then you could publish very quickly. Okay. And then they proposed this base pairing specifically as a mechanism for copying DNA material. The rest became history. Okay. In fact, it's quite interesting that if you open Nature journal from decades ago, like even 70s, you see actually 2.5 times more papers than they publish now. Okay. Actually, they publish fewer papers nowadays than in the past, because nowadays papers became much longer. You have to have more figures to convince people that what they have is right. And whereas in the past, it used to be just a letter, right? It's called letter to editor, right? Letter to the editor. And you just write a letter, the editor, if the editor likes it, the editor publishes it. And sometimes you have a figure like this, most times you don't. So I read a Nature paper written by an Indian Nobel Prize winning scientist, Raman. Have you heard of Raman? Raman scattering Raman effect. And he won the Nobel Prize for discovering the Raman effect. But that paper was published in Nature in 1930 something. So I read that paper, and there's no figure, okay? Not a single figure. There's only one number. That's the focal length of the lens he used. But he won the Nobel Prize for that. Anyway, so life was very different back then, 60 years ago. So in terms of dimensions, you want to be familiar with this. Okay, it doesn't have, okay. Diameter is 20 angstroms. Can you see? Okay. And in fact, if you include some other molecules that are condensed on the DNA, probably effective diameter is a little larger, maybe 30 angstroms, but physically, it's just DNA alone is about 20 angstroms. And 10 base pairs make one turn, about 360 degrees, more or less. And then each step is 3.4 angstroms. In our cells, in our cells, we have 46 chromosomes, 23 from your mother and 23 from your father, right? So 23 chromosomes is about 3 billion base pairs. And then times 0.34 nanometers. Okay, so if you do the multiplication, it's about 1 meter of DNA per chromosomes you got from your mother. Same for your father, right? So in each cell of your body, you have 2 meters of DNA. So remember that. Okay. Do you know how many meters of DNA you make before you die?
You're going to tell us, aren't you?
Okay, how far can you go if you connect them back to back? To Chicago?
I'm going to say to Saturn.
Saturn. Okay. Why Saturn? You like Saturn?
I don't know. I was thinking outer planets, ridiculously long amounts of DNA. So, I make my own body mass in ATP every day.
Okay, the answer is 1 light year.
We're still orders of magnitude too small.
1 light year. Okay, 1 light year. I almost forgot the light year is a unit of length, but it is, right? How far the light travels in 1 year. And it is approximately 10 to the 16 meters. Okay. Okay. It largely comes from the fact, 2 things, that we have 10 to the 14 cells in our body. Okay? And then, some cells are made more than once. Okay? So on average, if the cells are made 100 times, then you get that. Just mainly the fact that you have a lot of cells. So I gave a talk and then in Aspen, and I mentioned this, and I got an email from a woman who works at a bank as a clerk, and she had, oh, that's really amazing. What about like, you know, volume? If you actually measure the volume, how? And it turns out volume is maybe a gallon, like milk gallon size. So actually, it's not that impressive if you count it as a volume, because the DNA is so thin, right? So if you want to impress people, just talk about the length, not the volume. DNA has charges. So on the backbone, you have a minus sign here. So you have a phosphate group that is charged negatively. So for every base of DNA, you have a single negative charge. So another way of showing this, you have DNA, that's the phosphate, and this is the sugar ring, and that's the base. So there's a single unit of DNA, and then okay, phosphate, sugar, and base. And I also have translation for the students from China. No, it is in Korean. And then you form a polymer of single-stranded DNA, and then when you form base pairing, then you get double-stranded DNA, right? So the color is actually all artificial. It's not. There's no real color here, but you have adenine and thymine as A and T forming a base pair, and cytosine and guanine also forming a base pair. Now, have you heard that the C and G base pair is stronger than AT base pair? So, okay, now if you stop somebody on the green street, okay, any student, okay, why is CG base pair more stable than AT? Okay? Then the answer would be, well, because between the two, you have 3 hydrogen bonds, and AT, you have 2 hydrogen bonds. So is that the answer? The answer is actually no. You'll be wrong. Okay? It's not that. It's because the real answer is that hydrophobic interactions, that's more specifically stacking interactions. So what are stacking interactions? That's why I brought my textbooks from my college years here. So I have a stack of books. Okay, I have Feynman lectures, and then I have quantum and classical, whatever. So these are the adenines and guanine. Okay, these are larger ones. Okay, so they, in terms of base, so A and G, they are large. They have 2 rings, and C and T, they have 1 ring. So they are, one is large, the other is small, and these are called. And if you combine the two, you know, as a group, you call them purines, and this is called pyrimidine. And I can tell you that one of the I's is a Y. Okay, okay, maybe this one is a Y. Anyway, the way I memorize this is that, okay, whatever is larger has a shorter name. Okay, that's how I memorize it. But anyway, so I have purines, I have pyrimidines. Okay, the way the base pairing work is that I have, you know, the big one and small one, they pair like this, right? That's the base pairing. And then now I have another base pair coming in, and then but they come in, and they make a 30-degree angle. I mean, a 36-degree angle like that to make a right-handed helix. Okay, come, help me. Okay, hold it at. But then they cannot see. Okay. So that, and then I come with a third base pair, and, oh, okay. Oh, okay. Then I come like that. That's the third base pair. Now, you have base pairing interaction, but what's more important is the hydrophobic interaction. These bases are hydrophobic. They have aromatic rings. They don't want to be in water. So by actually maximizing stacking interactions between the bases like that, flat against flat, by forming this interaction, they can maximize the hydrophobic interaction to avoid water. Okay? So this is actually the force that's driving DNA hybridization to form double helical structure. Okay? Now, so now if someone asks you on the street, then your answer is, okay, no, it's a stacking interaction that's doing this. But why still cytosine and guanine more stable? Right? That's because actually, on average, stacking interaction between C and G and other base is actually stronger than just nearest base. When people measure this adjacent stacking interactions, when that stacking pair includes C or G, you generally have a higher stacking interaction. So it's not really due to the base pair interaction, but because of the stacking interaction.
So if you have a single base pair of AT and a single base of CG, then the breaking would be...
That will never form, right? Because basically then you are exposing both phases to water, and they don't want to do that, right? So it's not easy to solubilize single base in high concentration in water because of this reason. Any other question? All right, so I'll see you on Tuesday.